Hey, Mr. P here. In this video we're going to talk about metabolism, and we're going to focus our discussion on how enzymes, through a variety of steps or stages in both kind of linear chains and cycles, come together to really produce this really complex system of interacting steps and biological processes. Okay, as this picture indicates, there are really large, complex interacting cycles and chains that all work in synergy in order to produce the ATP and produce the products that all organisms need in order to survive. Okay, so metabolism is really the sum total of all chemical reactions that occur within you as an organism, but also cellular metabolism is the sum total of all chemical reactions that occur in the cell as an individual unit. We know from previous lessons or from previous videos that cells are the basic unit of life, and so anything that is related to you as an organism really also is related to cellular structure and function as well. Within the umbrella of metabolism, there are two kind of overarching or two kind of subcategory or two subcategorical reactions. One of them is catabolic reactions and one is anabolic reactions. I know in previous videos we've kind of highlighted exactly what constitutes an anabolic reaction and what constitutes a catabolic reaction. I know we've looked at specific examples of both catabolic and anabolic reactions. Remember, anabolic reactions are the types of reactions that are going to build up larger complex or larger, more complex molecules. An example of this would be dehydration synthesis. Specifically, dehydration synthesis is the type of reaction that is going to link monomers in order to form polymers. Now, I know down here I say photosynthesis is an example of an anabolic process. Dehydration synthesis is a smaller type of process or a smaller type of reaction that occurs several times within this big umbrella reaction that is known as photosynthesis. Remember, photosynthesis is specifically a plant cell's, and even more specifically a chloroplast within that plant cell's ability to take carbon dioxide and water, two things that plants absolutely need, and in the presence of sunlight and chlorophyll will produce glucose, C6H12O6, and 6O2. This is the photosynthetic equation. We know from this equation that we're taking smaller compounds and we are building them. Specifically, we're taking the carbon and we're taking hydrogens and oxygens, building up larger compounds known as glucose. Okay, so through the process of photosynthesis, and this video obviously is not in any way trying to teach you anything there is to know about photosynthesis, other than this kind of idea that metabolism is a part of photosynthesis, or photosynthesis is a part of metabolism. Anabolic and catabolic relationships or reactions are a part of metabolism, as well as a part of photosynthesis. Cell respiration is another part of metabolism, which is more of a catabolic reaction, and I'll talk about that in a minute. Photosynthesis, though, is using smaller compounds to produce larger molecules, and glucose really, when we look at the photosynthetic equation, is a rather large molecule. There are 24 atoms, whereas there are only 3 atoms in this particular molecule. And I guess I should finish this out by saying 6 CO2 and 6 H2O come together to produce 1 glucose and 6 oxygen molecules. Glucose molecules are produced using this anabolic process. Carbohydrates like polysaccharides and proteins and lipids and all of these types of organic molecules are going to be produced by this anabolic process known as dehydration synthesis. Two terms that I'd like to introduce you to right now is endergonic and biosynthetic as it relates to anabolic processes. Endergonic means that your products are higher energy than the reactants. And so if you look at this graph down here, the endergonic reaction, we have point A and we have point B. A is obviously the reactants, and B are the products. And so if you look at the y-axis, this is the amount of free energy, which is really the amount of energy associated with that particular point in that particular reaction. And so as the reactants start at point A, they're going to require an input of energy. That's the only way we can actually increase the amount of free energy. If we need to get to a point where we have higher energy than we started, we have to add energy to it. It's the only way that works. So in an endergonic relationship or an endergonic reaction, we're going to increase the amount of energy by absorbing energy to a point where the activation energy, and we're going to talk about that in a little bit, but so that the activation energy is achieved and therefore the products can be produced. Now, where these products end in relation to where the reactant started is obviously higher on this free energy axis, and so we know that the products are higher energy than the reactants, and so therefore that is an endergonic relationship or an endergonic reaction, also called an endothermic reaction. Okay, another name. Endothermic or an endergonic reaction. An example of this would be biosynthetic, so more complex molecules are produced. Again, we have a more complex molecule, or a polypeptide is a more complex molecule, or a polysaccharide, like starch, cellulose, or glycogen, are produced as a result of dehydration synthesis or these anabolic relationships. So more complex molecules are produced as the result of a dehydration synthesis, which often produce or always produce higher energy products, known as an endergonic relationship or an endergonic reaction. And again, an example of that is photosynthesis. On the opposite side, catabolic reactions are the exact opposite. Catabolic relationships or catabolic reactions is when we break down complex molecules. And so if I wrote out the cellular respiration equation, we would see that C6H12O6, or glucose, plus 6O2, we breathe in oxygen, yields 6H2O plus 6CO2 plus energy. Okay, now we're taking this very large complex molecule and we're breaking it down into multiple or smaller, less complex molecules. And so the products are going to have less energy than the reactants, which makes sense because we got energy out of this equation by breaking it down. If we break the reactants down, and glucose being one of the reactants, if we break the reactant down and we end with less energy than we started, then the only plausible explanation is energy has to be released. And energy is released in an exergonic reaction. This is also called an exothermic reaction. Okay, exothermic versus endothermic. Endo, I like to remember energy comes into, and endo kind of are equivalent. Energy comes into the system. Endo. Exo sounds like exit. Energy exits the system and is released. Degradative is a term used to describe complex molecules being broken down. Again, because the more complex molecules are broken down to produce less complex molecules, energy is going to be released. That is an exergonic relationship or an exergonic reaction. That is a degradative process. It is a catabolic reaction. Catabolic and anabolic relationships or reactions both kind of funnel into this idea of metabolism, which again is the sum total of all chemical reactions that occur within a cell and within you as an organism. Hopefully this makes sense. If we look at how metabolic pathways exist or how metabolism works as a whole, we have to talk about metabolic pathways. Metabolic pathways are the pathways that are created by enzymatic or enzyme-catalyzed reactions to get from point A to point B. Often, the metabolism processes or the processes that constitute or make up our metabolism are not a single step reaction. We have to produce step A point 2 and A point 3 and A point 4 and A point 5 in order to get to B as our product. Okay, so this is a product that we ultimately want. So I'll say it's at the end. This is the preferred product that we want as an organism to produce. But we have this, which is our reactant, at step A that we are starting with. Okay, so we're going to start with point A with this particular reactant, and this reactant is going to produce this product. And so this is a product just like this is a product, but you'll notice that the shape of this product is slightly different than the shape of this product. And so this would be enzyme 1, this would be enzyme 2, enzyme 3, enzyme 4, and enzyme 5. You will notice that at every step, the product that is produced at that particular step changes slightly. Every single one of these conformations, or these three-dimensional shapes of this protein, is altered in a slight way. And so enzymes are going to produce these products, which are intermediate. I'm going to say intermediate products, and these intermediate products are ultimately needing to be produced in order to get to our end product, which is ultimately the goal. And so these specific sequences that make up or constitute this metabolic pathway is going to result in the products we want at the end of this entire process. Okay? The enzymes required to catalyze every reaction in these pathways are determined by a cell's genetic makeup, meaning the genes that are associated with this particular metabolic pathway have to be turned on and off in order to produce the enzymes or produce the products in the proper order or sequence in order to get to the proper end product, which is the red product in this case. Okay, so how do these metabolic pathways really work in real life examples? I have on this slide 2 real life examples of metabolic pathways. One of them is a chain or a linear or has a linear fashion, and one of them is a more cyclical metabolic pathway. So we've got a chain and we've got a cycle. Metabolic pathways exist in both chain and cycle form, but the thing they have in common is that it is a series of metabolic steps, and those metabolic steps are mediated or controlled by specific enzymes at each step. So in glycolysis, and this is glycolysis, you do not need to learn the steps of glycolysis in this particular video at this part in the IB syllabus. You will need to learn glycolysis obviously when we get to cell respiration, but that'll be a future video. Glycolysis is a series of reactions that is in this metabolic pathway to convert glucose into pyruvate. Now, the reason we need to convert glucose into pyruvate is that glucose is not directly utilized to produce ATP. And so in order to get ATP, which is the obvious goal of cell respiration, one big process, cell respiration, which includes both of these steps. Glycolysis is step 1 of cell respiration, then you have the link reaction, and then Krebs cycle is step 3, if you count the link reaction as step 2. And so both of these are going to feed into each other, but are both metabolic pathways in their own individual right. So glycolysis is when we take glucose, we convert it to pyruvate through the use of specific enzymes, which I haven't actually indicated. No need to learn the enzymes, but I wanted you to see that there are several steps. All of these are All of these steps are intermediate products, meaning they have to be produced before moving to the next step. Glucose cannot be directly converted to pyruvate. Glucose has to be converted to glucose 6-phosphate. We do that by adding a phosphate. I'm not going to get into the actual biology of these steps. Just know that we are taking glucose and we're adding a phosphate to it. Then we are changing it slightly and we're going to add another phosphate to it. So now it's a slightly different sugar with 2 phosphates on it. We're not getting directly from this 6-carbon chain to this 3-carbon molecule in one step. We take very small baby steps, changing one specific part at a time in order to get from this reactant to this product in a very controlled manner, which is really important because if it is uncontrolled, then energy is released too quickly or too much energy is released, and the cell can basically explode from the inside, which is bad. Okay? Glycolysis is a chain version of a metabolic pathway. Again, just like glycolysis is a chain, the Krebs cycle is a cycle. We, again, have several intermediate steps. Acetyl-CoA is going to be basically the molecule that starts the process. The coenzyme A is going to leave, and the acetyl, which is a 2-carbon molecule, is going to attach to oxaloacetate or oxaloacetic acid. You can say either way. That oxaloacetate, with the addition of the 2 carbons, is going to change to citrate, which is our first intermediate. It's going to change to isocitrate, which is our second intermediate. Alpha ketoglutarate is going to be our third intermediate, and so on. You get to succinate, fumarate, and malate, all the way back to your oxaloacetate. And these intermediates have to be produced in that order in order to get from point A to point B, which in a cycle is the same spot. Point A and point B is your start and stop, but you'll notice that the purpose of Krebs cycle is ultimately to produce molecules that are going to produce ATP. Okay? Metabolic pathways can be both chains or cycles. How does this idea of metabolic pathway, enzyme discussion, enzyme review, and exergonic and endogonic or endergonic kind of relate to a specific reaction? Activation energy is a really, really important concept when we look at a graph of a particular chemical reaction. We're going to have point A, like I've said before, and point B. A are, the reactants. Okay, point A is the reactants, point B are the products, and in order to get from point A to point B, you'll always notice when you look at a chemical reaction graph, there's always this kind of hump that the reaction has to get over in order for the products to be produced. We call this activation energy. Okay, activation energy. Activation energy is the difference from where we start to the top of that particular hump. Now you'll notice also that there are 2 lines here. One of them is very tall, one of them is very short. This is activation energy without an enzyme. This is the activation energy with enzyme. And so what happened? The activation energy was lowered. That's the work of a catalyst. Enzymes are considered biological catalysts. Catalysts do 2 things to a reaction. They lower the activation energy, which you can see here. Okay, we've in fact probably lowered the activation energy in this particular reaction by 1/3 to 1/4. So enzymes lower activation energy, or I should say catalysts in general lower activation energy, and they speed up the chemical reaction. It's much easier for a reaction to travel this path as it is to travel this path. If you were in a car, it would require less time and less gas to climb this hill or to take this tunnel through this mountain range, so to speak. Okay, this would be much faster and would require less energy in order to take this path as opposed to this path. But this path is the path that would have to be traversed in order for that chemical reaction to transpire without the use of an enzyme. So why are enzymes so important for metabolism and this metabolic pathway? Enzymes are crucial because of this energy lowering and speeding up the chemical energy or the chemical reaction idea. Metabolism would require too much energy if we didn't have enzymes, and it would take too long if we didn't have enzymes. So we employ enzymes as a 3-dimensional structural quaternary protein in order to take our reactants, which are often things that we consume, and turn them into B, our products, energy, ATP, things that we need at the cellular level. With the use of enzymes, can lower activation energy and speed up the chemical reaction. It increases our efficiency, of the chemical reaction, and therefore increasing the efficiency of the cell and of the organism. Okay? Enzymes do not alter the proportion of reactants to products. We are not changing A or B. Notice we are starting and ending at the exact same point. We're not changing the amount of energy we had in the reactants to begin with. We're not changing the amount of energy we had in the products to end with. We are just changing the trajectory of, or the path of that reaction. We're lowering the activation energy and we're speeding up the chemical reaction as a benefit to the organism. So, mechanisms of enzyme action. What happens in this enzyme? I know we've already had enzyme lectures, or we've already had an enzyme lecture, and so I'm going to kind of review really quickly the steps of enzymatic action. And all enzymatic action starts with the contacting of a substrate to the active site. Now you will notice that this active site does not exactly match the shape of this substrate, but the substrate has to impact the surface of the enzyme in order for the enzyme-substrate complex to form. Once the enzyme and substrate, or specifically the enzyme's active site and substrate, contact each other, then the enzyme is going to actually change the shape of its active site to accommodate the shape of the substrate. We call that the induced fit model, which is slightly different than the lock and key hypothesis, which we've used to discuss enzymes as well, which basically says one lock has to fit one key specifically and exactly in order for the key, and in this case substrate, to work correctly. But this is a more correct way of looking at it. The induced fit model says that the active site is going to slightly change to accommodate the shape of the substrate. Once the active site changes and the active site becomes accommodating to the specific substrate, then it can bond, and a temporary complex is formed between the substrate and the enzyme. We call this entire thing the enzyme-substrate complex, which is really, really important. That forms, which is kind of the most important step when we talk about an enzyme actually lowering the activation energy and speeding up the chemical reaction and the production of the correct products. If the substrate doesn't bond correctly to the enzyme, then the products are not going to be produced because the enzyme's not going to be able to work. At this point, the enzyme-substrate complex has formed and the activation energy is going to be lowered, and the substrate is then altered. In this case, one larger double molecule, let's say maybe a lactose, which is a disaccharide, is going to enter a lactase, and I can't read that very well, a lactase enzyme, which is going to produce the two individual monosaccharide components of lactose, which would be glucose and galactose, which when the enzyme does its thing and the activation energy is lowered and the substrate is altered, then we have released energy because we have broken that bond. And the only thing left to do then is to take the transformed substrate, which is now the product, and release it back out into the environment. That's probably the intracellular environment. And now they are individual products. Now we have individual glucose molecules and individual galactose molecules. These individual monosaccharides can now be utilized by the cell to produce ultimately ATP, or energy. And that's in a sense a very simple metabolic pathway, or a very simple enzyme-substrate interaction. Okay, so the unchanged enzyme, now you'll notice that it is identical to what it looked like here. Okay, it hasn't changed. It can now combine with a new substrate and it can work again. These enzymes can work over and over and over. They can be utilized multiple times in a row. They can be recycled. It just increases again the efficiency of this metabolism. Okay, very efficient. So let's look again at real quick review. First thing that happens, number one, is substrate binds to the enzyme's active site, which is altered to accept the substrate. Number two, molecular bonds become less stable due to binding, and so that's why the activation energy is reduced. Okay, we are becoming less stable, we are releasing energy, we're breaking the bonds. We now lower the activation energy because we have broken the bond. There's less potential energy in that molecule because the bond is broken, and the products can be released. So 4 steps to kind of enzymatic action from substrate to products. Really, really important. That's where we're going to end it. Next video, we're going to talk specifically about enzyme inhibition. Bring your questions to class. See ya.